Good evening, children. Welcome to another practical assignment for today. In today's practical, we will be doing TS of blastula or blastocyst of a mammal. This would be under the section D, that is spotting. So on a new sheet, you will write down today's date, 19th of August 2020, the practical number in pencil because this will change later, then the object, to identify the permanent slide of TS of blastula or blastocyst of a mammal with the help of distinguishing characteristics and a well labeled diagram. Now, see in your course, the option is given the TS of blastula or the blastocyst of a mammal to be studied by means of a chart or a slide. So, we are adopting the method of a permanent slide so that when the school reopens, whenever it reopens, we'll be studying the practical with a permanent slide. So, let us see how to begin. Your work is very simple. Blastocyst, we know children, it's the structure like this with a cavity, fluid filled cavity called as blastocele, right? There are cells inside, we call them inner cell mass, and a layer of trophoblast or trophoectoderm, right? So this blastocyst, how we will study, we'll start studying like, you'll start writing like this. You will write the heading in black color, and the next would be identifying characteristics and the entire work in points. Remember, you are studying this for the spotting segment. So in your final examinations in practical, not more than 3 to 4 characteristics I'll suggest you to write because it's a simple diagram, though the council says only two. But from the beginning, children, I've been emphasizing that if one goes wrong, the other would be taken as the correct one and marks would be provided. So let us not just stick to dot two characteristics. A little extra while learning will be in your favor. But for file work, we'll write all 9 characteristics. So let us see how to go. The first point is, this is the slide of TS of blastocyst of a mammal. It is a two-layered ball of embryonic cells. The cells of the outer layer elongates to form trophoblast or the trophoectoderm. So these cells, the outermost, these boundary of cells, this is called as trophoblast or trophoectoderm. Remember, this I have already done in theory, and I'd explained you that this outer membrane has nothing to do with the formation of the baby or the child in the mother's womb. The actual baby or embryo is formed from these cells inside what we call as the inner cell mass. The inner cell mass gives rise to the embryo proper. It has a fluid filled cavity called as the blastocele. This cavity, this is filled with a fluid. The cells of the trophoblast provide nutrition to the embryo. So if this is not taking part in the formation of embryo, then what is this trophoblast doing? It is providing the nutrition to the embryo inside, that is under development stage. Now, the face of blastocyst to which the inner cell mass is attached is called the embryonic or animal pole. That means this side. You can see it shifted almost to this side. So this will be called as the embryonic or the animal pole, whereas the opposite side, that means this side, will be called as embryonic pole. So while labeling children, you may not label it, but you have to understand that the side where the inner cell mass is attached is the animal pole or the embryonic side, whereas the opposite end is the embryonic pole. The same thing, children, while studying the theory, I have already explained you in the stepwise formation. I'll just remind you, this is what you have already studied in the theory portion while we were studying the chapter human reproduction. So here you saw how the morula changes into blastocyst. You are studying this for the practical purpose today, right? That is the blastocele, the cavity inside, the embryonal knob or the inner cell mass that is collecting in one corner, and the trophoectoblast. So this is what is the study for practical work today. The work is very simple, children, and I expect you to write 7 points, simple points today itself, and a most simple diagram of all the diagrams in your file. right? So this is the work for today. Please complete it at the earliest, but your assignment date I'll be putting in the Google Classroom on 22nd, right? So today is 19th. You must do the work at the earliest and submit the work latest by 22nd. So that's all for just now. This is our practical number 10 today. Thank you.